fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range. Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction as laser light strikes the cell. The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar, is placed in front of the forward scatter detector. The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. The scattered light received by the detector is translated into a voltage pulse. Because small cells produce a small amount of forward scatter and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population, but such a graph only presents one-dimensional data. Next, let's look at side scatter. As we have already seen, a cell traveling through the laser beam will scatter light at all angles. Light scattering at larger angles, for example to the side, is caused by granularity and structural complexity inside the cell. This side scattered light is focused through a lens system and is collected by a separate detector, usually located 90 degrees from the laser's path. The signals collected by the side scatter detector can be plotted on one-dimensional histograms, like we saw for forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram is, in reality, multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots. You can see that the peaks from the forward and side scatter histograms correlate with the colored dots in the scatter plot. Now we can view the results obtained when we create a scatter plot using forward and side scatter data from a typical peripheral blood cell run. The populations that emerge include lymphocytes, which are small cells possessing low internal complexity, monocytes, which are medium-sized cells with slightly more internal complexity, and neutrophils and other granulocytes, which are large cells that have a lot of internal complexity. This multi-parametric analysis is the real power of flow cytometry.